Good evening, everyone, and welcome to our 5th Seeds of Knowledge webinar. I'd like to begin by acknowledging the traditional owners and custodians of the many lands from which we're tuning in today. I myself am joining from Wadawurrung country, and the seed bank is on Djadjawarrung country. Our guest speaker, Meg, is tuning in from Naarm, and the herbarium is located on Wurundjeri country, as well as the numerous peoples and countries over which we all travel to do our work and people who contribute to our research. I'd like to pay my respects to their elders past and present and extend that respect to Aboriginal and Torres Strait Islander peoples who might be attending this evening. By now, you know me, I'm Elle Fox, and I'm the Partnerships Coordinator here at Seeding Victoria. I'd like to acknowledge the funding from Bullock and Northern Grampians Landcare Network to help Seeding Victoria run this Seeds of Knowledge program. We love that we're able to have such a strong partnership with the Bullock Northern Grampians Landcare Network, and we truly appreciate the continued support. If you're interested in becoming a member of Seeding Victoria, please feel free to email me at info@seedbank.com.au and we'll have a chat. It is free to join. I'd like to introduce Dr. Meg Hirst. She doesn't know this, but she's someone who I find truly inspirational, and her work is fundamental to botany and seed ecology. So I'm super excited to hear all about your work tonight. Thanks, Meg. I'll pass over to you.
Oh, thanks, Elle. My name's Meg, and I just wanted to share with you some projects I'm working on with my gardens colleagues, as well as folk from Deakin University in the plant ecology lab there. So before I start, I just wanted to acknowledge First Peoples knowledge systems, and I recognise that the Western system of botany isn't the only way of knowing and understanding plants, algae and fungi. So to begin, seeds can be overlooked, especially next to those beautiful floral displays, but under magnification, we can truly appreciate their hidden beauty. And it's seeds that allow plants to move through space and through time. And it's with that sense of wonder. And their amazing ability to lie dormant for 10 to hundreds of years, which provides a way to ensure against extinction of plants in the wild. However, all around us, plants are under threat as their habitats are lost or degraded. So more than 85% of Australian plants occur nowhere else in the world. So of these endemic species, there is quite a high list at risk of extinction. So this is an alarming indicator of the health of Australia's biodiversity. And pictured here, we've got the nationally endangered Borea mirabilis from the Grampians, the Grampians pincushion lily. So this is known as a resurrection plant, and as you see that beautiful bright orange foliage, that's an indication that it's starting to dry down. And it looks like fireworks. But this species is an extreme example whereby the population is only one genotype in a location, but nevertheless, you still need a framework to assess it in order to protect it. And because of that, we look at all these threatened species in Victoria that are under direct threat, and we need strategies in place and significant conservation work to safeguard both individual species and their plant communities. So from the Botanic Gardens here, we work on an array of threatened Victorian plants covering plants, fungi, allied organisms, and we investigate, research and develop species recovery plans and actions along with our partner organisations. And so this leads me to the first thing I wanted to talk about today. I'm just going to introduce 4 different areas I'm working on at the moment. And this one is preventing the extinction of Victoria's threatened flora. So this is a collaborative, as you can see with all the names there on the title page, a collaborative project based here at the Gardens, bringing science, conservation, horticulture and on-ground managers together to halt the extinction of 24 endangered or critically endangered Victorian plants. And the way we do that, we have to have this integrated framework, what we call an integrated conservation framework. And our project addresses these key species recovery plans to optimise conservation actions across the life cycle of these 24 species. And this will grow. We will add more species as we go. However, we need to focus on these key knowledge gaps. So what we've got is what we consider 8 activities, and I'll just click through them, that makes up our framework. So you can see from survey work to optimising germination, particularly with species that are difficult, collections, conservation genetics. Then we look at the pollination studies, ex situ, developing ex situ living collections, and that's along with our conservation horticulturalists, establishing new populations, and then of course disseminating findings through community workshops. So just getting the knowledge out there. And so I just want to use an example of how this has worked. And this is part of our strategic framework. And this is the Grevillea jephcottii, the Pine Mountain Grevillea, and this is a nationally endangered Victorian endemic. Now, this is before we started on our 24 species, and it was just as the 2019-2020 bushfires were happening. But we were looking at this species purely because of its endangered status. But before we could really get into it, 95% of its range was burnt out. So because of that, we needed to come up with this framework, and again from those 8 activities we've chosen 4 that were really the most suitable to find out what sort of support we could do for conservation management that would contribute really meaningfully right at that moment in time. So knowing there were 2 unburnt populations remaining, we needed to assess this real concern that another fire might come through, and of course in that case would deplete the soil seed bank, and then any post-seedling emergence and then establishment. So this roll-on effect. Looking at the different strategies that we had, the first was that we had no seeds banked in the Victorian Conservation Seed Bank. So at one of the 2 unburnt sites, we were able to collect 20 seed, 2 of which you can see there in the picture. And so we couldn't attempt any seed banking protocols. We couldn't obviously optimise germination. And so it became clear that we really needed to monitor remaining wild populations and make quality ex situ seed and living collections. So this is how we went about it. We had to develop this ex situ living collection to act as a kind of an insurance for the short term while we worked out what was going to happen post-fire. And so to assess this, we needed to assess the genetic diversity and structure of the 13 wild populations, and that would inform our collecting plans. Population geneticists, Tara and Laura, went out and took leaf samples from adult or seedlings, depending on whether the sites were fire affected or not. And the genetic analysis revealed 8 geographically defined genetic clusters. So this is interesting because it actually shows us how to effectively and efficiently collect for species and conserve genetic diversity from each of those 8 genetic clusters that were identified. So knowing this, we were able to prioritise the sites in which we needed to go out and get cutting material. And so we did that from multiple individuals across those geographic clusters just to maximise the representation of the species genetic diversity. And this is really important going forward. So we share this information with on-ground management, and we're mindful that we build our ex situ living collections, but at the same time we need to monitor the wild populations. So this is where we've used this integrated approach with both on-ground and ex situ collections. So far this is working really well, this integrated framework, and this is part of one of our big ones with the preventing the extinction of Victoria's flora. So moving from Pine Mountain, we're going to the Bogong High Plains, and this is a collaborative project I'm working on with the Extreme Ecology Lab. I love the name Extreme Ecology. That's Susanna's lab in Deakin at Burwood. And here we're exploring the germination niche of selected alpine species, and this is to build a greater understanding toward a seed-based approach to alpine restoration. As in any goal in seed-based restoration, it's the successful transition from seed to established plant. However, this is obviously can be challenging. Seeds form the primary means of reproduction and species persistence for many flowering plants, and they have different recruitment requirements. And so these differences on how they produce and disperse, it's really critical. So we need to understand what we're working with and what plant communities we're working with, and also especially under the effects of climate change. So one of the issues we have is for germination to occur. We know that species have specific requirements and that they've evolved a range of dormancy and germination timing mechanisms to sense that external environment and germinate during periods which we would consider that are most conducive to seedling establishment. Not always the case. But so what we have to do is understand what each species may have of these specific requirements. And that's really important when we look at the Australian Alps, because here it's under pressure from warming and extremes with climate change, and there's a predicted shift in the composition of the ecosystems with reduced diversity and function. So habitat degradation from weeds, feral herbivores, human activity, this further threatens this really fragile ecosystem. So seed-based restoration is one way of looking. It's less expensive and more logistically feasible as a method compared to, say, planting tube stock. Yet there's these challenges that limit its success. And so seed germination and seedling performance, they can be prone to high rates of failure in the alpine environment due to these harsh microclimatic conditions. So seed germination is temperature sensitive, so making the persistence of populations and colonisation of available habitats vulnerable to warming. So although there's direct seeding technologies that offer the potential for large-scale application in these very rugged and at times remote areas, the success rate is actually extremely low. So going with that, and this is the paper that we've just submitted, there's evidence of insufficient seed globally to support large-scale restoration efforts, and I'm already probably talking to people that are very well aware of this. But there's the fundamental research on the recruitment niches and germination requirements are really needed to improve the efficacy of seed usage. So in this study, we've taken a focus on the thermal germination requirements of 20 Australian alpine species that are reasonably common within a community. Up the top left there, you'll see the map of Australia that shows where the alpine region is, and the accompanying box is showing Mount Baw, and then the larger map is showing the Bogong High Plains. And next to the map are 20 beautiful seed images taken from the wonderful Bob Hare, who is our science volunteer at the gardens. So very, very big shout out to Bob. He just takes some wonderful images. So we collected these seeds from at least 10% of individuals of the same population, and then we pooled them. We chose not to cold stratify seeds in this particular study, and a lot of them would germinate without this treatment. But we wanted to sort of present a technique that could potentially simplify the restoration process. I can go through the methods, but basically we sowed 7 replicates across a temperature gradient, and up the top there, that is what you see from the purple going through to the orange is basically going from the cold across to the hot on the thermogradient plate. And this is where we position our Petri dishes, which are actually very, very small. They're only 35 mil, and there's 196 of these. So we have them all color-coded and so forth. But I'm happy to talk another time about methods. But I just wanted to just show you some basic results going through with one species. So using Leucochrysum alpinum, that beautiful summit species, if we look at this, the germination, and this again is on that thermogradient plate, so you've got a temperature banding from say 5 going up to roughly 35 degrees. And so germination takes place between what we'd consider a minimum and then a ceiling temperature, with the highest germination rate used to define what we call the optimal temperature. So if you looked at that green rectangle shaded space there, that's the thermal threshold for germination. So we can see that the Leucochrysum alpinum germinates between that breadth. So we're basically saying it germinates between 14 to 29 degrees. And if you see the dashed line down the center, that's telling you the optimal temperature. So it sits at around 20.2 degrees. If we then incorporate the mean soil temperature for where the seed was collected, and in this case, False Creek, and we modelled these temperatures where we use 50% germination rate with the number of days per year when the soil temperature reaches the germination thermal threshold. So basically it will tell you that the snow-free days are about 132 where this species can actually germinate in the field. And then we quantify this by then saying, okay, well, that's maybe the current situation. What if we calculated using 2 IPCC global change scenarios? Would we see any differences there? So we calculated the number of days per year when the maximum soil temperature reaches the thermal threshold for the climate scenario above or below. And so here you've got your blue bar, that is the suboptimal, and it's associated with germination efficiency, so basically non-stressful germination. And then the orange is the supra-optimal, and it predicts a more stressful type environment. And so what we do there is we look at this as an actual framework again, I guess, as a picture of how this species may perform now and into the future. So we can then say that Leucochrysum alpinum is unlikely to be reduced by future climate scenarios. So it's still doing really well. So if we put it all together, we have a summit species. So up on Mount Nelson, for example, we have a summit species with a very wide thermal germination niche. It germinates across 5 of the 7 temperatures that we tested. It has high germination rates and optimal temperatures for germination to occur during a significant proportion of snow-free days. So when we looked at our 20 species, they all behave in a way that we could then categorize. I'm not going to spend a lot of time on this, but basically we can start looking at, if you put your attention again to that green the rectangle space, the shaded space, that's our thermal germination niche, or a GNB, the germination niche breadth. And you can start seeing that just visually, just some of the differences from the available days, that snow-free days for optimal germination to occur, across to just how species may behave now and into the future, with some species actually increasing their likelihood of persistence. The way we can visualize this is with a principal component analysis. And again, not going into this too much, but if we looked at all our 20 species, we can actually start saying that from this data that we pulled together, 70% of them are really good candidates for seed-based restoration. They have high germination rates, wide germination niches. They germinate through a snow-free period, extended, and there's opportunities if we look under future warmer temperatures. Again, we have candidates that they performed well, but they still need attention for the future. They didn't perform as well in the higher temperatures. To then see ones that are specific requirements, so they're really tailored, where we need to actually look at what we can do, because they have increasing opportunities in the future, but they did not perform well. And then, of course, those that have conservation actions required, and that is in our D category, of which they are all listed taxa, so threatened taxa. And again, that is where we then can put our efforts into conservation actions. So if we were to look at this for implications, from what we looked at, we found that the mid-spring is best to sow your seed and when the soil moisture is high. We found that you would have to tweak that for species that had really narrow thermal thresholds, and that would be combining that again with the soil moisture data, finding those specific times, and then whether you decide to postpone again, if it's a predicted dry summer, to go in when you've got the greater rainfall later on. So we found that some species deviated from general patterns and that there was variation, obviously. Also, we didn't do the cold stratification, as I said. Maybe that would also expand our germination window. So these are areas that we are still looking at. And it takes a collaborative effort, obviously, to do any kind of seed-based restoration. So we're really targeting how we would approach that with on-ground managers. So that was Falls Creek, Bogong High Plains, treeless. We're now subalpine, and this is about the Wittsteinia vacciniaceae, which is the bauble berry. And again, this is working with some of my dear colleagues from Deakin. So the bauble berry. The reason behind this work was to really highlight the role of conservation horticulture. It's an emerging discipline driven by botanic gardens working towards global strategies to address plant conservation. And there's often an emphasis on ex situ living plant collections. As I mentioned, like the Grevillea, jephcottii, they're of high preservation value. So considered a conduit position, the role brings together the ecology of the species and the horticultural techniques to successfully propagate it and provide subsequent plant material for ex situ living collections, reintroduction, or just more broadly into cultivation. So building information on how to successfully reproduce threatened plant species is critical to understand how species may persist in different environments and plant communities. And again, especially if we consider climate change. And of course, species range sizes, whether they're expanding or contracting. So the bauble berry, we thought this was a really good species. It's a strong candidate to test conservation horticultural methods. So it's a vulnerable subalpine species with a limited distribution to a few mountainous areas of Victoria. So hence it's an endemic Victorian species, occurs nowhere else in the world. It's in the family. Now I need to pause here because I can never say this. And if there was a taxonomist in the audience, I would ask them to unmute and please say this family name, because anyway, I'm going to attempt. So the family is Alpinum, and it's known for its ancient Gondwana flora. And it's fundamental to safeguard this species against any potential evolutionary loss, considering just how unique it is. So fire frequency and intensity have been deemed high risks for this species. It has no effective post-fire response strategies. On top of that, it is at risk of, it's compounded by the added likelihood, I guess, of the habitat becoming too dry. And it is also the breeding microhabitat of the critically endangered bauble frog. So these frogs are endemic to the Bauble Plateau and are Victoria's only endemic amphibian. Basically, the bauble berry is located in rainforest margins, sheltered subalpine woodland and shrubland, growing against tree trunks and large boulders. It's mainly found between Lake Mountain, Donabuang, and the Bauble Plateau. There's a couple of isolated occurrences, Mount Cobbler and Wilson's Promontory. And my colleague, Dr. Lee Hassan, he applied some environmental niche modelling just to explore if you were exploring new sites outside of a population's range. And although it's contentious in this case, because the populations are healthy, there is no need for any translocation options. We just wanted to test that environmental niche modelling to look at what actually would come up. And it was interesting that one little area of Cape Otway, of course, we still need to consider microhabitat. To get the material to test for cultivation success, we accessed 5 of the 6 known localities. Unfortunately, we could not sample on Mount Latrobe due to access issues, and that is in the pipeline. The only locality that was fruiting was the largest population on Mount Baw. So all germination work that I'm going to talk about is only from that locality. This is Danny White and Virginia Williamson just there, and that's at the Cranbourne Nursery, where we basically propagated cutting material across those different populations. But what we wanted to do, we wanted to test 2 very common propagation techniques, either using a mix or using plugs. And then we also wanted to know if you added the rooting hormone, which is a common application to initiate roots. So this is what we did. And basically we found there were differences between the 5 populations, Lake Mountain showing high root initiation across all treatments, but we had 60.8% root initiation irrespective of treatment. So that really said that this species roots readily. It's pretty easy to work with. From a seed bank point of view, that was a different story. So we had fresh seed, again, from Mount Baw, and we had to remove the fruit and cleaned it. We measured seed traits and x-rayed large samples. We did that because if you x-ray and you get a fill rate, it's easier knowing going into an experiment that you're only working with filled seed, where the likelihood for germination is obviously going to be greater than doing cut tests, which are quite destructive. I'm happy to talk about that. So as seen in the germination trials, we incorporated the plant hormone gibberellic acid. This is not ecologically meaningful, but it's great lab work if you want to bypass issues such as a physiological dormancy, which is the most common dormancy type. But before testing the thermal threshold, we really needed to know if this species was going to germinate to begin with. And so we did have a really good response in our standard testing. But we wanted to know how it would behave across a germination band. So we set up 1,960 filled seeds in 2 treatment groups. Basically, if you remember the thermogradient plate I showed you before, the same one, you've got your 35 ml plates, 196, and they're across the whole grid, running from 5 to up to 35. So when we look at the germination here, we can see that, again, although not shaded in green, which would have been useful, here the germination breadth for the bawbawberry berry is between 8 and 22 degrees Celsius, with an optimal temperature sitting at around 15. That was great because we picked that up, that it germinates in the standard conditions, which is 15 dropping to 20 degrees Celsius. So it was good that we were able to pick that up. Often one of the issues when we do standard testing, if we have species that are very, very discrete in their narrow niche for germination, we can miss it in just our standard testing procedures. So that was good to know. So again, just thinking about that paper we did on the alpine seed restoration, we can explore this with the bawbawberry, and here by incorporating the soil temperatures using loggers where the seed was collected and overlaying that germination niche breadth, so you can see there it's in green on the soil temperature graph, we can see that around October in Mount Baw, there's a higher chance seeds will germinate as the temperatures begin to rise and soil moisture is high due to the recent snow melt. So these are times are kind of consistent with natural seedling emergence. Seeds could also be sown in late autumn before the winter snow and receive natural cold stratification. And again, as I mentioned, this could potentially expand the germination niche threshold and increase your germination rates. but there is a real risk there that the seed could be lost. We observed seedlings in April at Mount Donnabuang where the seeds had clearly been transported downstream and germinated in a washout area under leaf litter. So we do know there is a lot of movement of water. So from a conservation horticulturalist or for a budding one, this provides the background of how we now know we can germinate and work with the bawball berry. We know it has a reasonable thermal germination need, and it's best sown either in mid-spring for restoration success. We also know that it relies on a soil seed bank for reproduction, so we need to monitor that. The use of gibberellic acid may increase the germination window in studies. However, germination will take place without it. And similarly, we can exploit the natural adventitious root formation. So you don't need root hormones when applied working under nursery conditions. So we can now grow ex situ collections from either seed or vegetative material. And we can keep them containerized or we can grow them in garden beds. So just to conclude on that, the species requiring these conservation actions, we've now tailored what we would say is part of that framework where you're adding all this additional information. And Polytechnic there, with you see the young lad holding some plants, they're propagating the Wittsteinia for us because it's a great way to talk about conservation horticulture and trying to get an understanding of why you might be sampling particular populations or holding or have holdings of populations more than one within a nursery setting. So we need conservation horticulturalists to strengthen and disseminate our findings. And so this is a really important area that I'm actually incredibly passionate about. But from conservation horticulture, I now go to the conservation generation. So this is children of the present and future, as we know, will bear the cost of climate change and therefore must be involved in climate decision making through climate literacy education. So the persistently high number of threatened species and the continuing addition, well, we want them to be added sometimes, we need to keep pushing on that, indicates a need to reimagine how we might approach conservation. And it's a timely question to whether existing programs are working and are they sufficient enough to address current global challenges, and whether we need to increase our level of community engagement and understanding to support greater change. So to address this level of concern in public awareness and engagement, we're working on a program, Raising Rarity, and it's to actively engage people in local plant conservation. It's an outreach program and it is a collaborative venture, and basically it's trying to get plant conservation outside of the Botanic Gardens setting because we're experiencing we are experiencing a progressive loss in human nature interactions. So the less we experience, the less we know, and the less likely we are to contribute or to champion for threatened species. So this is what this program is about, and it's a school-based program. So one of the things that was interesting is that from our reading it's kind of individual actions like, turning a light bulb on and off. They're potentially less effective because of a perception that they're insignificant in the face of climate change. So really we need this collective action to foster a powerful sense of hope. And so this is what we're trying to do through our school program, Raising Rarity. And it embeds conservation, education and action into a hands-on approach. So the program has recruited students to grow and care for a threatened species that occurs or did so historically within the proximity to their school. And the Raising Rarity School Partnership begins by the students attending this Discovery Day, which is hosted at the Gardens. And this is where we introduce students to the plant that they're going to grow. and they get up and close with it. They get the microscopes out. We basically put up a description, a taxonomic description, and the students work through it to get an understanding of what they're looking at. And from that, so they've got a sort of an understanding about the species. We then go to their school where we establish research beds. I'm calling them research beds, but we plots, and the kids plant the species. I'm going to say Grevillea canis at the moment because this is 3 pupils from Rosedale Primary School, just there on that path. And so with the Grevillea canis, they then plant them, and that is our research bed establishment. And then it becomes a data collection phase. And that's where the students, along with their teachers, measure phenology through observation. So they look at vegetative time to flowering, and then time to seeding. And that's just through using particular codes that we can then look at. And that gives us that really great way of looking at how plants perform under cultivation, and whether we're going to see differences in flowering time, or because these kids water the plants, they take the pH, they feed them if they need to. And then we have seed collection, where the pupils basically have put little baggies on the seed heads, collect the seed, and then they come to us, which we call Celebration Day. And last year we celebrated at Royal Botanic Gardens, and we had Rosedale Primary School, Locksport Primary School, and some students from Glengarry, and they came and we celebrated this harvest, so to speak, and it was just a wonderful experience. So we're calling it Raising Rarity. It's Raising Rarity with schools. Currently, the program's working with 6 schools on 2 threatened Victorian species. We're increasing student engagement. We'll be working with 10 schools this year, and then up to 16 the following year. And that is through some wonderful philanthropic funding that has just come in just recently. So we're very grateful for the support in this program. So Rosedale Primary School has been one of the first to take off, and you can see the students there. There's Matilda in front of her Grevillea canis plot, all beautifully flowering, the grey billy button. And what these students, they embraced it so much and just loved working with this species. Up the top corner, you'll see a group. Now, some of those are the wonderful field naturalists from Latrobe Valley. So the Latrobe Valley field naturalists, we were doing a community survey, and of course we were looking at areas where the school, it's just clicked over. So I'm going to have to, no, I'm going to go back. Sorry, I'm going to go back because I want to say this. So the students had the opportunity to join us at a national park it's a local national park, with members of the Latrobe Valley Field Nats, who are amazing and bring chairs, sandwiches, drinks, coffee. Like they know how to survey, and they came, and so the kids were part of it. They had an opportunity to come out and look in these where there were historic recordings. They learned how to use a GPS. Unfortunately, they didn't actually need it because we didn't find any. But the point is, they were part of it, and that's something I think that. That's really important to me anyway, just how we've got this wonderful field naturalists that embraced the kids coming along, but they got to come to the school because the plants are in flower. So they get to see what it looks like without having to go, because where it naturally occurs is actually on a regional railway line. So coming to the school is probably a safer option. And it was just this wonderful way of all combining. Early results, they have a school mural now where the billy buttons is prominent. The students in year 6 have it on their senior tops. So engagement, I would say, is high at Rosedale Primary School. That's how we bag the seed heads. From a research perspective, I would normally bag only 1 flower head per bag. But here the kids are involved, and we're doing it as a collective cultivated collection. So we also test it, again, on the thermogradient plate. Although that's a reasonably low germination there, we cut it quite short. That was only a 2-week window. We're also looking at how this seed may behave for going forward. So imagine this is kind of where we're at. if schools really came involved. And as I said, we had 6 schools last year, 10 this year, and then we'll build to 16. What we really want to do is build these school hubs where they all support each other. And that would probably be involving a botanic garden within that hub, like we have involved Sale Botanic Garden, for example. So we're now in the planning stages of another regional hub. That pretty much is some of the things I'm working on. I hope it made sense, but also because I'm just going to be really, really cheeky here and just say, come to Horsham. I don't know, come to Horsham, please, to the wonderful conference that is all about seed science, and it'll be fabulous.
I'd also like to say come to Horsham, because Seeds of Vic will be there and we'll be supporting, and we'd love to see everybody there as well. Thanks so much, Meg. I think the biggest question that came to my mind straight away, and I was getting texts from people as you were talking, how can we involve our networks or groups in the Raising Rarity project through our junior Landcare, especially?
Yes,
especially,
that would be, yes.
Yes, and I think it's very timely because I think Landcare Victoria Junior Landcare and Biodiversity grants just opened today and close on March 28th. So what is the best way to potentially, yes, another question asked about TAFEs as well, potentially if they could get involved.
Yeah.
So I think everyone is very keen to see how we can help with this, because this is an awesome project. I'm just getting all excited listening to it.
And look, it's taken a while because we've had to, you've got to first, it's all about teachers, because the teachers have to be on board because they're overworked anyway. But you get a passionate teacher and there'll be success. That's one thing just we're working out, that you need to have a level of engagement already. And so with Rosedale, and we've also got metropolitan schools involved as well, we've found that really if they're already wanting to be part of it, it's just made it so much easier. And creating these hubs, it just makes sense because ultimately it's all about the plant. So you're looking at the species distribution, and then you can match. So you match where the primary schools are, where the botanic garden might be, and then you can kind of create these hubs. But as you say, it can be beyond that. It doesn't. That's just a framework we're beginning as we build, and that we now are beginning to get some funding, which is because we've got support from the gardens, but we need additional funding, and that's just been fantastic that we've just got some.
We've had a lot of questions about potentially if private nurseries or even like Landcare groups can help with the Raising Rarity with their junior groups or anything like that. I think I'm wondering if the best way is if you send me some information, Meg, and
Sure.
If people are willing to give me their email addresses, I can then forward that information to them with how they can potentially become involved with the program or support, or what to apply for if they're going to apply for funding via a grant.
Sure. We do have Raising Rarity at rbg.vic.gov.au.
So another question we had was, are you working with councils and shires to help with these plant conservation projects?
Yes, we are. And tomorrow, actually, I'll be in Sale with Wellington Shire, just talking about how we can look at some plantings in some new spacings that they've got that have come up. And that's through already having a relationship with colleagues from Sale Botanic Gardens. We've then been able to meet, so Justin, who's an environmental officer at Wellington Shire, we'll be going to meet with him and just discuss opportunities where we might be able to look at putting plants in some of the areas that he is under his watch. And the beauty of that is that he and his team can also monitor, and that, of course, anyone that's involved in any kind of plantings, the big question is, but who's going to monitor it? And so that's because that obviously can lead to huge failure rates if there's no backup on,
Yeah.
So that's an important one. But yes, we want to do more of that, basically, because it's a community outreach. So it's basically trying to harness the enthusiasm or the drive that people have, and trying to just see where we can go. But starting with, for me, I'm working with the school groups, and expanding across to who I meet along the way. Like the amazing field naturalists. I can't tell you how impressed I am when they come to surveys. It blows me away.
The wealth of knowledge just hidden away in people's minds. It's amazing.
Oh, I know. And they know more plants than I will ever be able to know. They know all the birds. They know the bird sounds. It's just, you're just completely humbled.
The experts, and often they don't acknowledge just how much they're contributing and how much knowledge they have as well. They're just so underutilized and under-recognized, I think so as well. So we had another question that was a little bit unrelated, but I had a similar question, so I'm going to ask it anyway. When you're taking cuttings, how do you then store those cuttings when transporting them back from a rural area back to the Botanic Gardens?
Yeah, that's a really good question. So depending how long we're away, we take larger cuttings than we would cut to begin with. So we would take, depending on the plant, of course, we would then we would either soak them overnight and keep them in a bucket if we're away, or we would roll them in very, very wet newspaper and keep them in a bucket and take them back. Alternatively, if we've gone for a very long time, say a week, we may bring plugs with us, and we actually do it in the field, because that way you don't want that moisture loss, and you can keep them in their own little humidity kind of, you know what I mean? You can keep it in their own little thing. Yeah, I've got props everywhere. If you like, what do you want? But yeah, so basically it's to keep them turgid. You've just really got to make sure they don't lose that moisture. And so I go for larger, and then I cut down to the smaller ones. And I mean, this is a Exocarpus, and that was from Saturday. And here it's just like, it's doing okay just in the water, but couldn't really have that in the car. Maybe you could have it where your coffee holder was. It's more important, isn't it, than your coffee? Yes.
So I have a few questions that are based around translating what you've told us into practical on-ground things for us and Landcare people and people who maybe don't have access to a lot of these research facilities as well. And one of the questions was, is there a way to mimic soil temperatures in a nursery hothouse environment, or is it a bit futile if you don't have heat mats?
That's really interesting, because I used, if you're familiar with propagation beds, and they have the cables that run through.
Yep.
So you've got these heating cables, and then you've got your moisture leaf, so it knows when to irrigate. So what I do in a nursery setting, and people used to use it for root initiation on rose cuttings, for example, I used those propagation beds, and I ramped up the heating on it and took off the irrigation component, so the leaf wouldn't irrigate automatically. You can get them, I'm not going to plug anyone here, but you can get them from different sizes, and they sit very nicely either outside or inside, depending on your nursery setup. But you can ramp up the soil temperature and just put some loggers in. And if you email me, I can find out more about what some of the things that we've done from a nursery point of view, if you actually want some suggested methods that we've got running. But I've found those horticultural boxes, and they're not, I mean, I don't think they're that expensive, but I used to have a couple at home, put it that way. And you can really ramp up the heat if you add on some different sensors and so forth. But it's doable.
So yeah, I wanted to thank you again because especially, I mean, all your research is so valuable and understanding, the germination thresholds and all that is so important for us to know. You did mention a bit earlier about X-ray for seeds versus cut test.
Oh,
And
yes.
yeah, I was just wondering if you wouldn't mind maybe elaborating a bit on that.
Sure. When I first started in the lab, we did not have an X-ray machine. A lot of the facilities came through with the bushfire recovery. So prior to that, we couldn't assess the seed quality. So we would slice it open, which is a cut test, and we would look under a microscope for basically a beautiful white glistening, something that appeared to be really healthy. The cut test is destructive. And then the other one we used to do is TZ testing, tetrazolium testing. And again, that is destructive, but you're looking at staining to indicate, that the seed's respiring and so forth. From a practical point of view, cut tests, you can't go wrong. And you can, even if you I always take it, no, regardless, we'll take a small sample and slice through them. So one, I can see where the embryo might be, or I get a good understanding of what I'm looking at. X-ray is great for just basically getting your fill rate percentage, particularly for long-term storage, because again, a lot of the work we do is for trying to get seed down to completely clean, no debris, and then all this. The fill rate and then how it germinates and so forth, so we can map it and track it over time and if it declines and so forth. But the cut test, I think you should be doing anyway. You should do it in the field. Sometimes a bit hard, but if you have a hand lens, and depending what seed you're looking at, obviously, I mean, the field nets would probably have a sharp knife, wouldn't they, for their apples? So you could just, just go, hey, I just want to cut open some acacia seed, do you mind? Field, if you can do it have an idea in the field first, that's always handy, particularly with the high level of predation that you will bring back with you.
Yes. Definitely. We understand that pain.
Yes, it doesn't end with us.
No. I had another interesting question here. So, are there any consideration in long-term conservation plans within seed banks that also consider the protection and restoration of the specialised pollinators and fungi species that act as prerequisites for the plant species to sustain itself in the landscape?
Yes. So let's just go back to that preventing the extinction of Victoria's threatened species.
Yep.
Basically, if you look at those conservation frameworks, there's pollination studies, there's rhizobia studies, and basically the background information, because you can't put a plant out if the pollinators aren't present, for example. Yeah. So yes, but again, it's costly. If we actually look at these frameworks, and they, as I said with the Grevillea, you come down to what you can actually do and what you can support when there's one limited seed, for example. So what do you do? Well, then you try and safeguard the genetic representation across, what I'm saying? But all of that's costly. And you're housing them in a nursery that might be under-resourced. And so, yeah, we are doing that. We are looking at the bigger picture. And of course, it all comes down to, again, those interactions. So yes, we are doing that.
Yeah, cool. That kind of leads into one of the first questions was what exactly goes into a pollination study?
Lots of camera activity.
Oh, that's cool.
Linda, who works on this, she's actually doing all the pollination studies. And she basically does a lot of trapping, a lot of filming. Then she looks at the seed quality, looks at the floral parts to understand. Linda did actually a really, really good talk at the last ESA conference. And if you send an email, maybe we can get that talk and send it back to you, because it will be saved somewhere on what she does in her pollination studies and what she's come across, and unexpected pollinators.
Yeah.
So, which is really cool because it's all on film.
That is really cool. That's exciting.
Yeah, I know. And anything Nushka does is super exciting with the orchids. So
I know, right?
Yeah, I know.
So I have a question here about, I'm just going to read it out. Okay. With there being so many threatened species, is there a way to communicate with local landcare networks what they should aim to incorporate in their plantings, and with the state government restrictions on threatened species, i.e. taking cuttings and seeds? I guess, how can we get that info, and how can we get DECA on board with enabling conservation activities? For example, the Yarra Gums range expands to Central Highlands, but we can't do anything with them to actually get more out into the field.
Yeah, that's a really good point. And that was something we were talking about just today, about movement and trying to understand what falls under the regulations for example, in translocation activities. Again, I suggest, if you really want to get more information on that, is to talk to David Cantrell and Nushka, who oversee our side of what is happening with the prevention of that project I just mentioned earlier. I think they are the people within our organisation who talk to DECA and all the relevant bodies, because there are so many moving parts. I am but one little wheel over here, but
yes.
But I do appreciate there are so many parts, and the movement of plants, of course. Let's look at our cultural history on the movement of plants, and we and the botanic gardens with the cycads, for example. We are very mindful of where we sit and what our responsibilities are, and so, yeah, look, it's a big issue. It's actually a really big issue. But as I say, with community outreach, I think we need to have a voice, because I know I want a voice. I want to be able to plant and restore certain areas that might be on private land, and how I go about that. So again, I think talking to people that actually talk to the bigger wheels is probably the better way to go. I wish I could answer that.
Yes, I know we've had
I'm sorry.
No, it's a hard question, and we all have that same question, and we all have varying levels of success in getting our voice heard, and it is an issue, like you've said, and it does need to be addressed, because we're all saying something and it feels like no one's listening. I think we'll wind up there. Thanks so much for coming tonight, Meg, and everything was so amazing, and especially because the Seeds of Knowledge program was devised as a way to really get this research and knowledge out to everyone, and it being free so that everyone can hear about it. And I know everyone's getting excited about raising Rarity and getting included now, so yay, that's exciting.
It is. It's super exciting. I just think it's just so nice if we can kind of take some time and keep these conversations going.